All right. So the way these work is I'm here to answer your questions, and I will make them available to everybody, obviously, but there's nothing new or anything that, that I'll say here that wouldn't be available to everybody anywhere. So that's what's up. Who wants to start with a question? No, you had a question. You want to ask a question?
Yeah, sure. What is dabzil chloride?
What is dabzil chloride? Where'd you get that from?
It was in the midterm.
No, it wasn't on the midterm. It was on an old exam, right?
Yeah.
Well, what's my policy on old exams?
It's not on the pages.
So, first of all, I don't answer questions on that exam that I gave you. And the reason that I don't like students using old exams is they waste their time with things like this. Have we talked about dabzil chloride in class?
No, that's why.
So then why would you waste any time on it? Okay? So seriously, I mean, I find students are fascinated with old exams, and old exams, the reason I don't provide them other than that one is to give you format. And exams change from year to year. And there's so many things that are on there that it's much more useful for you to spend your time worrying about the content and not what's on an old exam. Okay? So I always tell the story. I didn't tell you guys this year because it gets boring, but I'll tell you the story here since it's come up. That I was author on a real life textbook back in about 2000, and so I prepared materials for that for students to have practice questions and things like that. So over the span of 1 month, I wrote 6,000 questions. Okay, if you do the math, that's about 200 questions a day. All right, so that's why studying old exams, there's no way you're going to study old exams and stay on top of things. Okay, so the most important thing isn't that you get used to the questions. The most important thing is you get used to the content that's in the course. That's where I want you to place your emphasis. Okay. Okay. Other questions? Sorry. Yes?
Can you review Kcat?
Kcat, okay. So let's talk about Kcat. Kcat sounds like something a veterinarian would need, but okay. Kcat. So where are we at? Here. There we go. So Kcat is a parameter that we use to measure enzyme velocity, okay? And Vmax also measures enzyme velocity. So I don't want to say that, for example, Vmax does not measure enzyme velocity. All right? It does. Okay? The problem with Vmax is it measures the velocity of a reaction in a tube, but the velocity of that reaction is dependent upon how much enzyme is in that tube. So if I double the amount of enzyme, I double the amount of velocity. Just like if I double the number of factories, I'll double the number of cars being produced per time. Okay? So Vmax is useful and it's needed for other parameters like calculating Kcat and for determining Km, but it's not useful for comparing enzymes. Okay? I had a student earlier today ask me why that you could use Kcat to compare enzymes, and I said because it's independent of anything having to do with concentration. It's a characteristic of an enzyme, just like Km is a characteristic of an enzyme. So I can compare Kms of enzymes and say, okay, this enzyme has a low Km, this enzyme has a high Km, all right? So this one that has a low Km has a greater affinity for its substrate than the one that has the high Km. So, and I can do the same with Kcat. This has a high Kcat, this has a low Kcat, so this guy has a faster velocity than the other one does. I can't do that with Vmax because if I use one amount of enzyme for one reaction and a different amount for a different reaction, then it's really dependent upon what's in that reaction, that is how much enzyme I used. All right, so that's the background on Kcat. That's why we want to have Kcat. Now, your question, I think, also was, how do I get Kcat? All right, so Kcat is a parameter that is actually derived very easily. In fact, it's trivial once you know what Vmax is. So the way I would determine Kcat for an enzyme would be to do what I said in class, would be to take, say, 20 tubes of material. I'd put the same amount of buffer in each one, the same amount of enzyme in each one, and I would put varying amounts of substrate in each one and let each one go for the same time period, which would be a fairly short time period because I want initial velocity. Each of those tubes is going to give me a different velocity based upon how much material, how much product it produces with time. And if I look at this bottom plot, that's very much what I would see. That is that it would go up fairly linearly at first, and then it would level off. And it's leveling off because the enzyme is getting saturated with substrate. That is, that as soon as one product is released, a new substrate is available to bind in there, and there's no gap. Right? So that's what saturation really means. So doing this, I could generate data. I could either estimate from this plot what Vmax is, or I could do Lineweaver-Burke and determine a precise Vmax from the 1 over Vmax intercept on the y-axis. Okay, so that would give me Vmax quite accurately. Then, since I use the same amount of enzyme in every tube, it's a trivial thing then for me to take that Vmax value that I determined and divide it by the concentration of the enzyme. When I do that, then I will have finally the Kcat value. Okay, and the units of Kcat, as I said, are numbers per second. So let's say I said 50 per second. That would mean 50 molecules of product per molecule of enzyme per second. And the reason that that turns out to be that way is I had, if you remember, that velocity is measured in terms of concentration per time, concentration of product per time. If I take concentration of product and divide it by concentration of enzyme, the two concentrations actually cancel out, and so I'm left with number. And that's number of product per one of enzyme per time. Does that help? Sort of. Okay. Yes, sir.
I may be wrong, but I was reading a book. I think I read that Kcat was actually K2, which is when when the intermediate becomes the product. So Kcat does not represent the whole reaction, does it?
So Kcat, I'm actually giving you some simplifications. His question has to do with some other kinetic constants that relate to velocity of reactions. And you're right in that what is done in Kcat are some assumptions moving forward. Okay? So there are forward-moving constants, there are backwards-moving constants. You're talking about a forward-moving constant versus a backward-moving constant. Ideally, we're deriving only that forward-moving constant in the short time frames that we do that. But there's always a consideration with that, yes. Okay? Yes? Mm-hmm. Kcat's absolutely a way to compare different enzymes. So I can compare a DNA polymerase with a protease because each of them are producing a product, and each of them are producing a product per time, and each of them I can measure how many molecules of product per time per enzyme I can make. So absolutely I can compare different enzymes with that, you bet. And that's why we want to have it, just like I can compare Km. Yes? No, the graph I would make would be this one, because I just described to you they all have the same amount of enzyme. Okay? In this case, they're measuring time versus product. Okay? And we haven't talked about time versus product reactions. This is a velocity versus substrate concentration product. Okay? Ultimately, yes, you would do that, but this is a more complicated consideration, so I'm not talking about that. Yes, Naveed. Yes. Okay, so how much should you know about anything? You should know as much about anything as I've talked about it.
Okay, so you talked about the chemical system, for example.
Right. In that lecture, I actually said what you needed to know of that, so I'm not going to repeat that here. But the basic rule is what I talk about in class is fair game. Okay, so I'm not going to go and pick obscure things out, but in that particular lecture, I did say which of those you needed to know in terms of substrate. It's in the lecture. Yes, sir.
Can you go over cation and
Cation and anion exchange. Okay, so when we're thinking about various types of chromatography, there are several of them that we went through. And one of those was what's called ion exchange chromatography, of which there are two types, cation exchange and anion exchange chromatography. And so if I pull that up, okay, they rely on the fact that molecules that we're interested in separating sometimes are charged. If they're proteins, that's not uncommonly the case that they would be charged. And so they'll have a net charge associated with them. And if we similarly use packing material, the beads that I talked about. If we use packing material that has charged groups on it, and we arrange this properly, we can actually separate molecules on the basis of their charge. So the two types of ion exchange chromatography that we have are cation exchange chromatography and anion exchange chromatography. Okay? So let me just pick one. I'll just pick cation exchange chromatography and go with that. So let's imagine that I say, okay, I'm going to do cation exchange chromatography. So I get some beads and I pack them in my column. The beads that I pack have a little sphere, many little spheres, each one of which has a negative charge. Okay? So the negative charge, as you can see on the beads right there. As I tell students, we can't simply put negative ions into something. It's not possible to do. I can't just put chloride ions into something. I put sodium chloride ions in there. Notice I have a sodium and chloride, right? So I can't just put negative beads in here, even though that's what we show. When I put those beads in there, they have counterions. And those counterions, of course, are going to be the opposite charge. And they're usually something like sodium or potassium, right? So this thing starts out with little beads, and they're surrounded by sodiums because the sodiums are attracted to these negatively charged beads. All right? So I take a mixture of proteins, and these proteins that I am wanting to separate, some of them are positively charged, some of them are negatively charged, some of them are neutral, have no charge. Okay? So those positively charged ones will come here, and they see these negative beads, and they literally push the sodiums out of the way. So that's the exchange part. The sodium ions are being exchanged for a different positive ion. Right? So it's called cation exchange because what's being exchanged is the original cation, the positively charged sodium. Right? So in cation exchange chromatography, the things that will stick to the beads will be the cations. The beads themselves will be negatively charged. And it's reversed if I do anion exchange chromatography. There I will have positively charged beads and negatively charged counterions, like, let's say, chloride. Okay? So that's what's up with each of those. So if I do that cation exchange chromatography, what happens then is the positively charged ones stick. The negatively charged ones go racing out of there as quickly as they can. The neutral ones will take a little bit more time to get out. And the last ones will be the positively charged ions that are there. Okay? And then it's reversed, of course, if we're doing anion exchange chromatography. Yes, ma'am?
So if you have two versions that are positively charged, how would you tell the difference between them? Would it be based on their length?
So if I had two different proteins that were positively charged, I wouldn't be able to separate them by this technique unless one were really, really positively charged and the other one were barely positively charged. Remember, we use techniques in parallel or in series to purify a protein. So I wouldn't rely on this just as the way to get my protein. This would allow me to separate the positively charged ones from the negatively charged ones, but I would use other techniques that would allow me to separate something where they were both positively charged. Maybe I could use a gel exclusion chromatography where I've got big versus little. They're both positively charged, one's big versus the other one's little. I could separate them that way. Right. Oh, this could be a second step. I mean, there's not a particular order of these. Yes?
Covalently bonded?
Are these covalently bonded? Absolutely not. Because if they were, we'd never get them off the column. Okay? So these are attracted by ionic interactions with each other. And the way we get them off the column is we add sodium. We actually reverse the process. And the way we do it is we just add a ton of sodium. Sodium chloride, of course. We add a ton of sodium chloride to this. And the positively charged sodium will displace the proteins because we put a high enough concentration, they just outcompete them.
What do you do with the negative? You said you put chloride.
Uh-huh.
Chloride there. What do you do with the chloride itself?
What happens to the chloride? Well, remember that positively charged proteins can't come off by themselves. What are they going to come off with? Chloride. So I never have a situation where I get completely one or the other. That's just not possible to do. Okay? So that's what I get. Yes?
So you said the gel electrophoresis was for differentiating the size of the protein.
Yes?
But if I wanted to differentiate it by the charge of the protein, like how positive charge it is, what would I use?
Well, one, by how positively charged it is?
Yeah.
Okay. So there's a very good exam question. All right? You guys are better at making up exam questions than I am. Okay? If I wanted, what technique have I talked about in class that would allow me to separate something on the basis of how much charge it had? Isoelectric focusing. Yep. I should have thought about that question for the exam. You know, I have been known to go back and alter exams after I've written them, so there is still time. She's right. Yes. I'm happy to report there is an extra credit question on the exam. So, yes. So, you guys sang well. This class has been the best group of singers. I've been very happy with that. So, yes, I'm sorry, question.
So the 2D gel electrophoresis,
Yes.
can you go over how you would actually read the...
Okay. So, she's asking about 2D gel electrophoresis and how I would actually read a gel that I had done. So let's do that. I think these are pretty cool, as you probably gathered from what I said in class. And there is a schematic of 2D gel. All right. So we remember that the two dimensions, the first dimension is the separation on the basis of charge. So we have things that are really positively charged on one side, really negatively charged on the other side, and a gradient of charges in the middle. Okay? We separate them on that basis first, and then we take that sort of gelatinous column that they're embedded in. We add some SDS to it so that we're making an SDS page, and then we electrophorese that material from the top one into the bottom SDS page gel. And so in that second dimension, what's being separated is molecules on the basis of their size. And so we ultimately have something now that looks like this. So in this case, we would actually have the positive charge over here, so we would have the positive electrode over here, we would have the negative charge over here. So on something that was way up here would have something right up here would be large and negatively charged. Something over here would be large and positively charged. Something up here, large and neutrally charged. And then, of course, down here, small and neutral, etc. So we could actually look at that gel and in an instant tell where any given protein fit into that scheme, how charged it was, how big it was. And the beauty of the technique is that it is so good, it literally can separate every single protein that's present in a cell. And that's a really powerful thing, because now it becomes in a matter of analyzing spots and determining which spot between two different gels changes. So we have the cancer cell versus the regular cell. Which spot changes? Does nothing change? Does something change? If it changes, what protein is that? I want to know more about that protein. So it's a very, very powerful technique. Does that answer your question? Yeah. So it's a good question. So I've drawn it for you in terms of numbers of charges, but it turns out that the separation, and it's not inconsistent with what I said, but the separation is actually on the basis of pi. Okay, so where their PIs actually are the basis of how they separate. Because what happens is there's a gradient of charge that appears in there, and they separate by their PIs. So it's relating to pH in there, but don't sweat that. Yeah?
When you treat enzymes,
Yes?
so do they now have a new enzyme that's used?
Oh, good question. So if I take and I treat a protein with a protease, I'm going to break it, let's say, at lysines and arginines. Do I have a new C-terminus and do I have a new N-terminus? That's correct. So every time I break one, I create a new C-terminus for the previous one and start a new N-terminus for the next one. So absolutely right. So if I had, let's say, 3 places where I broke a peptide into pieces, I would have 3 new N-termini and C-termini. You bet. Well, so they would no longer be part of the whole bigger molecule. So now I would have to determine charge of each one. Right. Yes?
I remember you said that chromatography is a very powerful technique for purifying proteins.
Yes.
And I was wondering if you could explain that.
Okay, sure. So his question is if I could explain how affinity chromatography worked, and I'd be happy to do that here. So affinity chromatography relies on the protein's affinity, that is, its tendency to bind a particular structure. Okay? Affinity chromatography is very, very commonly used. It's a very, very powerful technique, especially if your protein binds a very interesting or unusual structure. So the example I gave in class was, let's imagine my protein binds to ATP. Okay? So I would take and chemically bond ATP to a set of beads, very much like the beads we've been talking about. And I would pack my column full of that so that ATP is sticking off. They have, I think, glucose, but it doesn't really matter. But glucose or ATP, let's say glucose, since that's what I have on the screen. So I would put glucose on those beads up there. And then my question is, can I pull my protein out of this mixture? Well, I've got this mixture of all the proteins in the cell that I apply to the top of the column. That's always what I've got to start with, because all I've done is I've busted open a bunch of cells. I've always got a mixture of all the proteins to start with. I pour those proteins through, and only those proteins that bind glucose will stick to the column. Okay? Now, if my protein were the only one in the cell that bound to glucose, I've just got a one-step method for purifying my protein. Well, it turns out that glucose is a pretty common molecule, and there are a fair number of proteins that will bind glucose or glucose-like structures. Okay? So it won't be pure with this particular example, but there are places where it may very well be pure. So if I said ATP, I probably would have even more proteins that bind, because there are many, many, many proteins that bind to ATP and use ATP. So the ATP, if I use that as an example, I would definitely have to do some other purification techniques. But the beauty is that when I ran this column and I pulled off the things that stuck to the column, I would have every ATP binding protein in the cell. One step, I've got all of them. That's kind of cool, right? How would I get the proteins off the column? Add ATP, right? Give them what they want, they come off, and they all come off bound to ATP. It's kind of cool. Yes, ma'am?
Do you have like a problem using them then after that because you have them all bound to something else?
Oh, is there a problem with the fact that they're bound to what you've eluded them with? Yeah, that's a very good question, actually. So you've eluded them off this column, and now maybe you wanted to study the protein without ATP. So we could imagine, for example, that I want to do X-ray crystallography. So X-ray crystallography allows me to determine 3D structure of a protein. And if I wanted to do 3D structure of a protein and compare the bound versus the unbound, then that means I've got to have a way to get the bound free of the unbound, right? Everybody with me? Okay. So I'd like you to think for a minute if there are ways, there are techniques that we've used in class that might help us in the process of separating ATP from a protein. What's that? Well, not ways to break bonds, although that might be doing it, but then those bonds might also be disrupting the protein, right? So I probably want to get my protein undisrupted. It's a little bit of a trick question, but it makes you think about something here. So first of all, have we talked about techniques that will separate a protein from a small molecule? Okay, what would that... What's that? Dialysis might, if we could get a dialysis tubing that would allow the ATP out. Okay, what else might? What's that? Jet exclusion. Okay. Now, the problem with this is that they're traveling through these things. Once they let go of it, they've got a chance to rebind it again. Okay. So ideally, the way that we would do this, you guys wouldn't. I wouldn't ask you this question on an exam, but I could tell you how you would do it. Okay. Think about the fact that this is not a covalent binding to the, between the ATP and the protein. So whenever we don't have a covalent binding, we always have this off-on phenomenon. I always want you to keep this off-on phenomenon in mind, right? So the off-on phenomenon means that there's a certain amount of the time it's going to come off, and when it comes off, I can treat it with an enzyme to destroy the ATP. So over time, if I waited long enough, now I would have the ATP disappearing. If I wait and treat long enough for that, and then I could get my protein ultimately pure of that thing it's bound to. Does that make sense? Yeah. So the off-on thing is really a powerful way for you to, a really important thing for you to think about when we have non-covalent binding. And we'll talk more about that later as well. Yes?
If that was like an ionic interaction, could you treat it with salt to remove?
If that was an ionic interaction, could you treat that with salt to remove that? It's a good question. You probably could, yes, but there's probably more to the interaction than just simply an ionic interaction. And one of the things that happens with salt, we haven't talked about it here, but people use various concentrations of salt to precipitate proteins. And so that's actually used in some purification schemes, so we might have some other effects that might happen as a result of that. But yes, that's entirely possible. Good thinking. Yes?
So do you have ones like in the first step there, you've got both the wheat and the protein count that glucose there, but if you didn't have glucose and you have something maybe larger that the protein needed to bind more around it, do you have ones that won't work with this because the beads are kind of in the way?
Oh, okay. So her question is, are the beads ever a problem in terms of the binding of the material to that? I can't tell you the answer to that. In general, when you make these, you're pretty much coating these beads with these small molecules. So I can't think of a place where that would be a problem, but I won't say that it's not. We're also here talking about fairly simple molecules. When we have more complex protein interactions, sometimes we could take a piece of a peptide and put it on there, and that could, I imagine maybe the pieces themselves might get in the way, so it's possible, but I wouldn't say it would be likely or common. Yes? Could you repeat the question? I'm not sure I understand it. So, no. So her question is, I think the question is, does Km change if you have, or does Km not change if you have competitive versus non-competitive? Is that a fair way to state that? Okay. So no, that's not true. So if we look at let's go back to enzymes here. We look at competitive versus non-competitive. We see that in the case of competitive, that the Km actually increases, apparently. All right? And that is shown right down here. All right, so, okay. So here is the plot, the Lineweaver-Burke plot of 1 over V versus 1 over S for a reaction in which there's no inhibitor, and here's one in which there's a competitive inhibitor. So we see when we look at that they have, in both cases, the same Vmax because they cross at the x-axis, which is 1 over Vmax, but they have different values of Km. Okay? So the different values of Km in the case of competitive means that the apparent Km of the enzyme has changed. It has increased. Okay? However, when we do the same set of reactions with a non-competitive inhibitor, what we see is this, and that is that they have the same Km, but they have different values of Vmax. Okay? I like showing this on a Lineweaver-Burke because it's much easier, which is one of the reasons people do Lineweaver-Burkes, to show you what these things actually mean. We didn't address uncompetitive. So uncompetitive is actually a little bit more complicated, and for a non-majors class, I just decided it wasn't worth you're having to mentally sweat it, because it's actually a fair amount more complicated. Yes?
Quick question about the slope. What is the slope of the thing?
Well, the slope is Km over Vmax, but I think if you start throwing terms out there like that, people just get mumbledy-pop, in their heads, and I don't think that tells me anything more. Okay? So I think if you know where the points are, I think you're in much better shape. So knowing the slope doesn't really tell you anything more about those. Eric? Yeah. Would I consider it to be a coenzyme? Okay, so his question is ATP a coenzyme? The answer is basically no. So coenzymes are things that will facilitate an enzyme catalyzing a reaction, but a coenzyme will not itself react. Generally, when when enzymes are binding ATP, they're using it for something, and so there it's actually a substrate. So are there enzymes that might use ATP as a coenzyme? I suppose there might be, but I can't think of any off the top of my head. But I don't have much on the top of my head either. So. Other questions? Yeah. Uh-huh. Oh, yeah. So boy, Eric wants to make this really as hard for you guys as he possibly can. So his question is, if we think about ordered binding of substrates, which is what happens when we have binding of multiple substrates by an enzyme, how does the binding of a coenzyme affect that ordered binding? And as you might imagine, what the ordered binding tells us, first of all, is that the enzyme has to have the right configuration. So if we had the binding of the first substrate affects the binding of the second one. That's what ordered binding means. Okay. So can a coenzyme play a role in that? The answer is yes, it could. Many coenzymes, though, you have to keep in mind, are covalently bound to the enzyme. It's not an absolute requirement. So only for those that aren't covalently bound might we consider that happening. But most coenzymes are actually covalently bound. Okay? Good? Yes? MALDI TOF. Okay, MALDI TOF mass spectrometry. MALDI TOF mass spectrometry is a fairly revolutionary technique that has gotten very popular in recent years. To give you an idea about how old I am in the dark ages, when I was working on my master's degree back in the late 1970s, I used to do a fair amount of mass spectrometry. And we struggled and struggled and struggled to determine masses of things that were a couple of hundred mass units, and we would hope we would get close to their mass estimate, and it was a very tedious procedure. Today, with MALDI TOF, you can estimate to the decimal places atomic masses of things that may be a few thousand in molecular weight, and you can do on the order of 6,000 or 7,000 of them a day. Okay, so it's really revolutionized what we're able to do. So MALDI TOF, which was the one I thought I had the figure. I should dig up a figure for you guys. I had to draw this mental picture for you. MALDI TOF involves basically an evacuated chamber. So we could think of a tube that has a vacuum in it. One end of the tube has an electrode. The other end of the tube has an electrode. So we have a positive and we have a negative. Okay? And right near one end, we have our sample where we can insert it into this tube. The sample's inserted in the form of a crystal, so that it doesn't just go flying off as soon as you put it in. So it's embedded in a crystal that you put in this tube. Okay? The crystal is activated by a laser. So the laser shines on the sample, shines on the crystal, causes the crystal to volatilize, and since the sample being analyzed is in that crystal, it too volatilizes. The volatilization causes the sample to gain a charge. And that's critical because that charge can be used then to drive the material from one, let's say it's a positive, charge drives, if you have a positive electrode here and a positive sample, it gets driven towards the negative. This is why the vacuum is important, because if we don't have a vacuum, it'll react along the way with anything that it hits that's gaseous. We have a vacuum, however, and so the attraction to the far electrode, all right, is determined really by the size of the molecule that's charged. So something that has a mass of 500 will travel twice as fast as something that has a mass of 1,000 if they both have one charge, because they have the same force that's driving the two of them. And so, like pushing a Volkswagen Beetle is harder than pushing a truck. Okay? The force that's driving this ion from one to the other is determined by its mass. So all I have to do to determine the mass is very accurately determine the time it takes to move from the volatilization event to hitting the electrode. And we can actually measure when it hits the electrode. So that's how MALDI TOF works. I wish I had. Sorry, I didn't have a figure for that. I should have looked that up. Yes. Okay, sure. here's the figure from the book. So, well, actually, I think everybody's seen the book, but I'll show. Let's see. Right there. So the smaller molecule will move faster because the same force is going to move a small thing easier than it's going to move a large thing. Okay, so here is the thing that I'm talking about. All right, and okay, so you can see right here is where the sample is inserted. Okay, we see the laser volatilizing it. We see it's actually focused. I didn't say that, but there's actually a focusing of these that causes them to move in a line towards the other electrode. And all we're doing is measuring how long it takes them to do it. That's the time of flight. That's the TOF part of that. Okay. The MALDI is called matrix absorption of light. I can't remember the DI part. But that's where the whole acronym comes from. Okay? Cool technique. Okay, thank you very much. Yeah?
Would that mean that you would have had to have found all their masses and then
Would that mean you'd need to know all of your masses independent of that to compare them? I'm not sure I understand the question.
like, so you're putting this sample in, and then it's separating the
Okay.
and there's different protein, right?
Okay. Okay. Well, I think I know what you're getting at. So what MALDI TOF tells you, first of all, is it tells you mass. That's all you get. Okay. So I think what you're talking about is identifying those proteins. And if you want to identify those proteins, so let's say you do this and you say, okay, here's a protein, and it's broken into 3 or 4 pieces. One of those pieces has a mass of 512.13. And another one has a mass of 689.66. Okay. And another one has a blah, blah, blah mass. Okay. Well, each of these is going to be fragments of a bigger peptide. And it's very easy to do the math and say, well, what combination of amino acids adds up to 589.13? You say, oh, well, that's cysteine, lysine, arginine, tyrosine, right? And so now you go back to your database and you say, which proteins in this genome have, within a small peptide, these amino acids? And which one has the amino acids in this one? And the ones in this one? And so you can put those together and say, well, the only one that meets that criteria is a DNA polymerase. I haven't asked you to do that, but that's how that type of analysis is done. Okay? And this is all done, not surprisingly, on computer. But the computer can actually do that analysis quite rapidly and tell you an unknown protein, what it is. And that's how we can do 4,000 or 5,000 proteins in a day with this kind of analysis. It's cool.
I laugh because I said not by hand.
Not by hand, not with a calculator. Yes, sir.
I have a quick question on, so you talked about a little bit about SDS that wraps around protein.
Yes.
I was asking myself, what disturbs the protein structure that makes it unfold?
Okay, so his question is, when I take and I add SDS to proteins to do SDS polyacrylamide gel electrophoresis, it causes the protein to unfold. Why does it unfold? I think that's your question, right? Yeah, it's a very good question. So the primary reason that it unfolds is because the detergent itself has a long nonpolar end. Excuse me. Remember that a protein has, that a detergent is comprised of a fatty acid that has one end that's very nonpolar and the other end that is either a carboxyl or, in the case of a detergent, something that's been chemically modified like a sulfate or something like that. Okay. So it's actually the combination of those that allows this thing to occur. Primarily what's happening is that we have a folded up protein, and we have a folded up protein. That folded up protein has on its inside, if it's a protein that's dissolved in water, that's dissolved in the cytoplasm, on its inside it has primarily hydrophobic amino acids. Those hydrophobic amino acids get disrupted because this new hydrophobic tail sticks in the middle of them and stops or destabilizes the thing that holds the protein together as a glob. So now the protein comes apart, right? When I say comes apart, I simply mean it unfolds. It doesn't come apart piece by piece, but it unfolds. So that helps the thing to unfold. As these interact with each other, so they're interacting with the backbone of the polypeptide chain, we've got these SDSs that are sticking out there, and they're all negatively charged. And they all repel each other. And like hairs that are all sticking out, that are all positively charged, they all form this elongated structure. It's a little bit like a woolly worm. Okay? And the negative charge is all holding the thing out. It's not perfect. There are some proteins that don't do this well, so you could think of some examples that don't. But it works really, really well for the vast majority of proteins. Okay? Good questions. How are you guys feeling about this? You feeling good? You feeling bad? What are you feeling? Stressed? Okay. So I think stress is one of the worst things that you can have for an exam. All right? That's why I really worry about students that convince themselves an exam is going to be bad, because they make it bad for themselves. I'm serious about that. I've seen this happen so many times with good students. So if you allow yourself to be psyched out by an exam, all right, what's it going to be like in the workplace? In the workplace? What's what? It should be easier? I guarantee it's not. I've been there and I've done that. Okay? So the easiest that you're going to have is here. All right? So if you feel like you can't handle this, I'm not saying that to be mean, but I want you to kind of keep a perspective of this. All right? That this is training. This is a step in becoming a professional. This is leading you to that next thing that you can be successful with. All right? And so learning to deal with those stresses, learning to think when perhaps, oh my God, right? The boss is looking over my shoulder, right? It helps you in that process. But stressing about things isn't going to get you anywhere, all right? And learning to deal with that stress will help you also as you get later into the job market. Yes, sir. Okay, so his question is, if I have hemoglobin in the T state and I get it into a really, really high concentration of oxygen, will it move to the R state? And the answer is, yes, it will. Okay? And that's because cooperativity is what's happening. Binding of the first one causes that slight shape change that favors the binding of the second, that favors the binding of the third, that favors the binding of the fourth. So all these are flipping the enzyme. That's actually changing the configuration of the enzyme. The binding of the oxygen is causing that to happen. So it's actually flipping it from the T to the R state. And I saw a hand over here.
So when the oxygen binds to the hemoglobin, it moves the iron 0.4 A's up, and that causes it to go into the R state.
Okay, so his question is, the binding of the oxygen by the hemoglobin causes the iron to move up by 0.4 angstroms, and that's what causes it to go to the R state? The answer is ultimately yes. Ultimately, yes. Yes? The T state has a low affinity for oxygen. That's correct. So how does it bind in the first place, right? How does it bind in the first place? Well, the T state still binds. Okay? I didn't say T state didn't bind. I said it's a low affinity. Right? So how do you overcome a low affinity? Increase the concentration. And where's the concentration of oxygen high? In the lungs. So in the lungs, you've got a lot of oxygen. It's overcoming that low affinity. And now it's favoring even more binding. So that's the loading phenomenon that I talked about. Make sense? It goes the other way when it goes out to the tissues. Unloading of some favors now they're starting to convert from the R back to the T state. Okay. Yes?
And how is CO2 and low pH stabilize the hemoglobin to T state?
Okay. So how does the carbon dioxide binding and the protons favor the conversion back to the lower affinity binding state? Let me get my hemoglobin stuff here. Okay, and let's see, carbon dioxide and pH, carbon dioxide affects carbamide. Okay, so at the molecular level, if we look at carbon dioxide, this is what's happening. Okay, this is what's happening at the molecular level. We have carbon dioxide that binds to amine side chains, makes carbamate ions. All right. How does it do it was his question? Well, I've just changed something that is either zero charge or plus charge, depending upon what's really all the other things that are on this R group, into something that now has a negative charge sticking off of it. What happens if I mess with charges inside of a protein? I'm probably going to make some conformational changes. That's my mechanism. Yes, Eric? Yes. Uh-huh. Oh, okay. So his question is, I talked about how RNAs can be denatured much more readily if I use mercaptoethanol. His question is mercaptoethanol something that we have in our body? The answer is no. So mercaptoethanol is a reagent that we use in the laboratory. And the other follow-up question to this is, are there ways that we can reduce disulfide bonds in our body, and do we ever need to? The answer is yes, we can, and yes, we do, and yes, it's very important. Okay? So we have compounds in our body that are small peptides that have disulfide bonds, or actually have sulfhydryls that can add their electrons just like mercaptoethanol does and release that. So they're known as reducing compounds, and yes, we can do that in our body. So, but mercaptoethanol is just simply a reagent, so is DTT, a reagent used to do that in the laboratory. Yeah. Sulfhydryl has no charge on it unless the proton comes off. If the proton comes off, then it's negative charge. So when I say sulfhydryl, generally the proton's on, but if the proton came off, it would be negatively charged. I'm sorry? What's a sulfhydryl? Sulfhydryl is an SH.
Oh, okay.
So cysteine has sulfhydryl groups.
I was reading the book and it was talking about the ping-pong mechanism, and
Yes.
it didn't represent it very well.
Okay, so he was reading about the ping-pong mechanism in the book, and the book didn't represent it very well, and can I make it worse? So I'll see if I can. Is that what you said?
Make it better?
Oh, better. Okay, you wanted it better. All right, we'll see what we can do. Okay, come on here. So the ping-pong mechanism is called double displacement, and let me pull this up here for you. Okay, so all this shows are the molecules. It doesn't show you the enzyme. And the enzyme, as I described in class, is alternating between two different states. All right? Two different states. That's all that's happening with the enzyme. The enzyme is either attached to an oxygen or it's attached to a nitrogen. So what it's bound, what it's attached to will determine what it binds to. If it's attached to an oxygen, it's going to bind to something that has an amine. And if it's attached to an amine, it's going to bind to something that has an oxygen. So if I have this enzyme, and this shows the reaction because this is showing the overall process that happens. When I add this enzyme to this mixture and I start with this and this, I get this and this. How do I do that? Well, the enzyme binds, let's say it starts out with an oxygen it's bound to. The enzyme binds first to an aspartate, and it gives the oxygen to the aspartate. Aspartate becomes oxaloacetate. From the aspartate, it takes an amine, and then to the amine, it's now got a nitrogen. It now binds to the alpha-ketoglutarate, takes its oxygen, and makes it into glutamate by giving it its nitrogen. And now it's got a nitrogen again, and goes back here and does this. So just sequentially going one after the other, after the other, after the other. Yes, sir?
So the enzyme doesn't necessarily start with oxygen? It could start with...
It can start with either a nitrogen or an oxygen. Okay? But the point is that this reaction is being driven by this swapping of all that's going on. We look at it from the outside, and this is what we see on the outside. Question over here? If I took alpha-ketoglutarate and glutamate and started with those two, then I would produce aspartate and oxaloacetate. Right? People say, well, it goes backwards. Yeah, it can go backwards. I'm talking about what I start with. So if I start with this and this, it's going to force the production of this and this. If I start with alpha-ketoglutarate, your question is a very good one. If I start with alpha-ketoglutarate and glutamate, all I have to have is one that has an oxygen and one that has a nitrogen. I'll produce the other two. Make sense? But let me just finish one thing with that. So we'll talk about this actually next term. These are enzymes called transaminases There are other enzymes that do ping-pong, but transaminases are great examples because they have these paired of molecules, and you can see the reaction happening with both those. Hand up back over here. Yes?
Yes, it's when it has attached to nitrogen, it binds to it. Is that what you're saying?
When it's attached to a nitrogen, it'll bind to an oxygen. When it's attached to an oxygen, it'll bind to an amine. Yeah, okay?
What was it start with?
I'm sorry?
What was the enzyme start with? Like,
It can start with either.
it was produced by
Doesn't matter. Doesn't matter. It can start with either one. Doesn't matter. Yes?
So this happens in the active site of the enzyme.
This is happening in the active site of the enzyme. That's correct.
So what about, since they're both sitting in there,
Uh-huh.
they're not the same shape, are they?
Well, that's, okay, it's a very good question here. So she says these things don't even look alike. So they're all binding in the active site of the enzyme? Well, let's think about the active site of an enzyme. All right? We think about that lock and key, and so we think we have to have a tight fit. But what we discover is that not all enzymes have tight fits, which means they have wide ranges of specificity. We saw, for example, that trypsin could work next to lysine or arginine. Right? So we can imagine that there's a little bit of flexibility in that binding site. Some enzymes have more flexibility than others, and guess what that flexibility affects? Km. Km. Okay? So it's a very good question. What's that? Well, if you have something that's very loose and very open to binding a lot of things, then it would probably have lower affinity than if it were very specific. Right? Cool. You can learn something with this. It's cool. Good questions. Very good questions here. What else you guys got? What's the secret to life, right? Yes, ma'am.
Can you talk about which forces that stabilize the different structures?
Forces that stabilize the different structures, okay? Well, we've got 4 different structures of proteins, I'm assuming we're talking about. Okay, so 4 levels of protein structure. We have primary, we have secondary, we have tertiary, and we have quaternary. When we think about primary, what's the primary structure of a protein? Amino acid sequence. So there's only one structure that's relevant there, and that is the covalent bonds that hold the individual amino acids to each other, right? When we think about secondary structure, how did I define secondary structure? Okay, so there was a very critical thing I said about secondary structure a lot of people miss. It's a regular repeating structure arising from interactions between amino acids that are 10 or fewer away. The regular repeating structure is important, okay? Now, for those type of things, we talk about hydrogen bonds because they help to stabilize because there's interactions between basically the rungs of the helix or the strand as we're talking about, okay? When we go to tertiary structure, we have a much more complicated set of interactions that are happening. So with a tertiary structure, we have things that are folded, and that folding can bring together groups that are ions. So we can have ionic bonds. It can bring together two sulfhydryls and make a disulfide, so we can have covalent bonds. It can bring together a binding site for a metal. You saw a metal today. You saw zinc being involved. Okay. It can bring together hydrophobic groups and make the hydrophobic groups help stabilize the protein. And it can have hydrogen bonds. All of those can help to stabilize the tertiary structure of a protein. All of those can also help to stabilize the quaternary structure of a protein, the quaternary being the interaction between different subunits. Okay? Yeah. Can I go over calculating the pH of a buffer system? What does that mean?
Like how you calculate pH if you have a weak acid, or
Okay. So let's say that I go back to here, and I've got a system that has, we'll do my favorite buffer, which is an acetate buffer. Okay. So I've got an acetate buffer. There's a problem that you work in the problem set that has starts out with sodium acetate, and to that mixture you add HCl. Now I find students, many students, confused to start with that, don't realize that you're starting with pure salt. That's one of those things I would hope you learned from organic chemistry, but you didn't, unfortunately. Your fault, their fault, I don't know, it doesn't really matter. But the point is that you're starting with a pure salt. So if you know the concentration of salt that you have there, and you know how much you add in the way of protons there, then the protons are going to do what when I add them to that solution? Not dissociate now. So what's going to happen? So if I have a salt and I add protons to a solution, what's going to happen? So the salt's going to bind to protons, right? Because it can. That's how a buffer works. So when I try, you need to remember this. A buffer does the following thing. A buffer is resisting a change in proton concentration. If I add something, it's going to resist it. How is it going to resist it? Well, in the case of protons, it's going to gobble them up. If I take away protons, what's it going to do? It's going to replace them. That's what resistance means. Right? How else would I resist it if I didn't do something about what I was adding to it? All right? So I have a solution that has, let's say, 0.1 molar sodium acetate, and to it I add 0.05 molar protons of the same volume. Okay, you notice that problem that I gave you in the book. All they work with were concentrations. They didn't give you moles, so you had to assume all the volumes weren't changing and so forth. So if I don't tell you that, you may assume that. And that's what they did there. So if I added 0.05 molar HCl, then that 0.05 molar of protons that I'm adding are going to get gobbled up by the salt. And so I'm going to end up with 0.1 minus 0.05 of salt left, which is going to leave me with 0.05, and it's going to leave me with 0.05 also of the HAC that I've now created, right? So in that case, I would say that the pH is equal to the pKa plus the log of 0.05 over 0.05. That's the log of 1, which is equal to 0. So there the pH would equal to pKa, and I could say that value is 4.76 because that's what the pKa is. Okay? So it's very important to understand what's happening in that solution. When I ask students, this is almost invariably what happens. I'll say, okay, you've got a buffer, and you add HCl. What's going to happen? And invariably, people will tell me the following. It's going to get more acidic. Well, I mean, that doesn't tell me anything, right? What's happening in the buffer? What's happening in the buffer is protons are going to get gobbled up. That's really the first thing that you should think of. What happens if I add sodium hydroxide? The pH is going to go up? No. Well, of course the pH is going to go up. If I add a base to a solution, the pH is going to go up. But I'm wanting you to understand how much it's going to go up. And to do that, you need to recognize that when I add a strong base to a solution, it's going to gobble up protons. It's going to take protons away. When that happens, the weak acid is going to give up protons to replace those that have been taken away. Very, very important concept. Okay? So if you don't have that concept, if that's not the first thing that you think of when you think of a solution or a buffer problem, you're going to have trouble with buffer problems. All right? You got to be thinking about what that buffer is doing and how it's doing it. It's really, really critical. Okay? Very fundamental to what buffers do. Now, I'm not working a problem in front of you, but that's the type of problem that I'm going to give you on an exam. Right? You're not going to have a calculator. Right? So I have to think of problems for you to work that aren't going to require a calculator. It means that you're going to need to know that Henderson-Hasselbalch equation well. What does that Henderson-Hasselbalch equation tell us? How can I use that Henderson-Hasselbalch equation to tell me pH? How can I use it to tell me if I need to pKa? How can I use it to tell me salt-acid ratios? Because each of those I can determine if I know the other two things. If I know the salt-acid ratio and I know the pH, I can find the pKa, right? And if I know the pH, I know the pKa, I can find the salt-acid ratio. And if I know the pKa and I know the salt-acid ratio, I can get the pH, right? So 90% of solving these problems is recognizing what you've been given and then how to plug them in and use them. So I take away the number part of it. You're not going to calculate any numbers. You're going to use equations. That's what you're going to do, all right? You will not have to crunch a single number on the exam. I promise you. Okay? Does that help, I hope? Okay. It's kind of hard without a blackboard up here, I have to say. So, okay, other questions? You guys are asking some very good questions. All right. I wish you the best. You have a question?
Classification of amino acids. So there's aliphatic and hydrophobic.
Yeah.
They both don't have ionizable partners.
Right.
So I was wondering what the difference between them besides just.
Okay. What I've just been asked is the most common question I've been asked this term. What's the difference between a hydrophobic and an aliphatic? Okay. And there's really none. That's just how they're grouped. Okay. So I've asked you to know the groups that are there. If I said, is something hydrophobic aliphatic, I would say yes. Okay? So all I'm asking you to know is what the book has given you in terms of those groups. You read different books, they'll have different words, they'll have different ways of organizing those. Don't sweat the subtleties of what that means. Just learn the groups that they're in. Okay? Oh, okay, now the questions are coming. Yes, quite back here. You can write in blood if you want to. As long as it's not mine, okay? Was there other questions? Okay, so good luck, study hard, and wish you the best.